Well, thanks everyone for coming. First of all, if for any reason you like tweeting about stuff as you see them, this is my Twitter account, so feel free to just tweet about stuff. So I'm going to talk today about the best research that was developed during my PhD 4 years. It's a lot, and I'll try to summarize it in a manageable way for everyone to understand. And it all starts with my interests, which are related to very different topics. So, bottom-up, top-down controls, species-area relationships, drivers of net primary productivity, organismal functioning. And these topics are related to disciplines that often do not talk the same language. They try, there's a disconnection between the way people approach different things. And I have always been curious about approaches that could try and bridge disciplines like those, for example. And one way, which is the approach I'm going to adopt throughout this talk, is a theory called the Metabolic Theory of Ecology, which is based on energetics, so based on the individual metabolic requirement, what are the constraints that propagate to population, community, and ecosystem levels. For most of the data I'm using is based on fish, mostly marine and freshwater fish. At some points I'm going to be talking specifically about reef fishes. So here we go. So the first step of everything is characterizing individual energy flux of fish. And that was something that we try to do. And I start with a very scary equation, but don't be scared about it. Basically, all this is trying to tell us is that the individual metabolic rate is a function of your body mass, and it follows a power function of this sort. This is our scaling exponent. The metabolic theory generally predicts this exponent to be 3/4. And it also follows an exponential dependence on the temperature, which at some point achieves an optimum of functioning, and then it drops until the organism eventually dies. So I'm mostly interested in estimating a couple of parameters to describe metabolic rates of fish, and those are the parameters highlighted here. Just noting that this denominator here, it's something we've been trying to develop for this particular paper, and it's not, although most of this equation is originally mechanistic in terms of kinetics and constraints of body size and metabolic rates, I have to recognize that this part is mostly empirical and I would say phenomenological. We don't really know the mechanisms that drive temperature optimum, but we do know its effect. Organisms, after some point, they just stop functioning. So this was a way we encountered to model that sort of curve. So to do that, we use data from fish-based metabolic rates. And to estimate the parameters, we used a Bayesian hierarchical model where we were estimating these parameters. And it's hierarchical because we partitioned the variance across different families. So we were interested in seeing how the families deviate from each other with respect to this, and this parameter here, and this one, sorry. So this is what we found, and which was really interesting because, as you can see, the 3/4 power scaling was well matched. This was all estimated from the data. The same thing with the temperature activation energy here. It's predicted to be something between 0.6, 0.7 electron volts, and we estimated from the data something very close to 0.6. So it's really consistent with the theoretical predictions. But very, very nice, and importantly, is this global optimum estimated to be something around 33 degrees Celsius. This is what the fitted data looks like. So you have metabolic rates corrected for a given temperature, and this is body mass. And the same here, we have metabolic rates corrected for body mass and varying across different temperatures. So it's very important to notice here that not every family has clear evidence of temperature optimum, and this is clearly a limitation of the data. But some families have very clear optimum curves being described here, for example, this one. But most alarming of all is this 33 global optimum here, which may be telling us that tropical organisms or tropical reef fish may be living very close to their temperatures, close to their optimum functioning. So this is yet to be studied. So as conclusions for this first part, we have size and temperature scaling that in agreement with theoretical predictions. We also have substantial taxon level variation in metabolic rates, which should also be telling us, given future directions in terms of trying to understand what explains this variance with regards to metabolic rates. And as I said before, tropical fishes may be experiencing temperatures close to respiratory optimum. Now I'm interested in understanding how these things constrain the abundance and biomass of reef fish across different scales. So I'll start with population. And for this particular thing, this particular section here, I'm going to deal with population abundance. So classically in population abundance ecology, we see people talking about factors like competition or a disturbance regime or habitat structure shaping the abundance of different populations at different sites. But we also have to recognize that ecosystems in general, they have energetic limits. So you have this scenario where you have a bunch of elephants in a small grassland. That is not possible because there's not energy enough to sustain all that population. So a more realistic scenario is something like that. So we do have energetic constraints on population abundance. But at the same time, we have energetic transfers going from top, bottom trophic levels to top trophic levels. If you're a big carnivore, you can only be sustained if you have a large amount of herbivores in the system. So there are all these constraints that are somehow shaping at the very limit those population abundances, but we don't really know the power and the predictability of those energetic constraints in dictating population abundance. And that's what we're trying to do here. So we start with the point of departure here is recognizing that species abundance distributions are generally characterized by having a lot of rare species and a few very, very abundant species in the system. So we first thought, well, so if you're very, very abundant in the system, maybe you're more likely to be energetically constrained. So we'll try and estimate how those individual level metabolic rate constraints affect the most abundant species in systems. The way this works on average, for example, is you have metabolic rates that scale with mass, as I showed you before with the 3/4 power scaling. And how does this relate to abundance? So if we consider an initial scenario where you have energy that is similar partition, it's the same energy partition across organisms of different size in a given trophic level, you would expect that the energy partitioning for each one of those populations is scaled to the -3/4. The consequences of that is just deriving a bunch of equations, is just telling us that total population energy use is independent of body mass. So this is the reason why this hypothesis has been called the energetic equivalence rule for this particular reason. And there has been evidence in favor and against. Is a very controversial hypothesis and the scale in which you deal with that. But yet at the same time, this is our point of departure. So we depart from this very naive, I should say, expectation that populations exhibit energetic equivalence. So the way we did this, again, we have an individual respiring, so this arrow here is just indicating fluxes of gas. And these are the curves that we would expect how those metabolic rates are functioning. So think of a place or an ecosystem where you have fluctuations in temperature across seasons. So it's warm in summer, then it gets cold in winter, and so forth. So the metabolic rate is changing through time because temperature is changing, and we know that temperature has an effect on metabolic rate. So this is an individual, and we want to scale things to population. So it's a simple matter of just summing across all the individuals in a given population, and then you integrate over time that fluctuation in temperature. So this is a very simple idea, but it can translate into a scary equation if people are not really used to do them, but it's actually very, very simple. I'm going to break it down. Well, before I break it down, actually, what I really like about this approach is that having a population level respiratory rate over time allows us to do a couple of tricks in terms of deriving predictions. The first of all is we can multiply and divide this equation by number of individuals, which is exactly the same thing, mathematically speaking. But the cool thing about this now is that we have an expression for the average abundance per unit area, which is the type of data we would collect in an ecosystem. You do, for example, a transect and you count the number of individuals. And at the same time, this also yields to us an average total biomass corrected by the metabolic rate capacity, average per individual, and then the seasonal effect. So this is basically what this equation is yielding in terms of predictions. We can have predictions for the abundance, how it relates to seasonality, to mass, and to temperature. If I do, I'm going to take a couple of jumps here, and I apologize for that in advance, but it's important. If we manipulate that equation, we get to this relationship where the abundance per unit area is related to the corrected biomass to the -1, and the average temperature seasonality to the -1 as well. So we're predicting from the theory that population abundance should scale to the -1 following those two predictors. Taking a second jump, we did an empirical expansion of this theoretical equation where we incorporate the effects of total species richness in a community and the area sampled in that particular community. And the reason for that is because if you sample more here and then you sample less there, you know there's an effect. You're going to have an effect on how much you're sampling rare individuals and so forth. So we wanted to account for that when we were actually estimating these parameters. And finally here, this is recognizing the limitation between trophic levels. So we would expect that after controlling for everything else in the system, you would see more herbivores than, for example, piscivores. I'm not going to talk about the other trophic levels in between because in reef systems they're very, very complicated and they eat a bunch of stuff and it gets really messy. But this serves as an initial departure in terms of predictions. So the way we did it, to tackle this question, looking at those predictors in terms of the most abundant species, we use quantile regression. And the data we used was a global, kind of near global database of reef fish community structure that I've been compiling with help of many collaborators worldwide. These are sites where we have encircled coral reefs, we have rocky reefs in squares, and then the different colors just represent different biogeographic regions. So because in these transects we estimate the size of each one of the individuals, an approximate size, we have to get at their biomass because that's what actually influences metabolic rates. So to do that, we use length-weight relationships published in the literature. This is just one example. To characterize for a given length what is the respective mass for that particular species. So we average the abundances across all the transects in a given site. So those 49 sites, they have abundances for each one of the species sampled in there. We consider 5 base level trophic levels, so just classic herbivores, omnivores, invertebrates, piscivores, and planktivores. Again, the reason for that is you can do this, or you can do something like 15 different trophic levels, and it gets really, really complicated to do with parameters and interpretations. So again, this is a simple, it's a simplification of reality that we adopted here. So the important thing that technically connects the first section with the second and the third section is the fact that we're propagating uncertainties. So when we estimate that parameter alpha, the mass scaling, and the activation energy, they have statistical uncertainties associated with them. And we have to bring those uncertainties when we calculate size-corrected biomass or average temperature, season kinetics, and stuff like that. So we used the full distribution of the parameters, if you kind of care about this sort of detail, to estimate these predictors, well, these two predictors here. So basically we used a mixed effect quantile regression, and the reason why we did it is because we wanted to control for unmeasured effects across the different sites. So our intercept was also varying according to the different sites. And the quantile regression approach is nice because it allows us to account for the effect of the different predictors going from very rare species to very abundant species. And that's what I'm going to focus now. And these are the results. So basically what this is showing is the effect of the trophic levels represented by different lines going from very rare to very abundant species. And these are represented by quantiles. So basically this is the effect of being a piscivore, going from very rare populations of piscivores to very abundant population of piscivores. That's the idea. So the interesting part here is that we do have a partitioning between low trophic levels and high trophic levels, so herbivores, planktivores, and omnivores up here, at the very, very abundant populations, to carnivores and then piscivores. I'm going to revisit this constraint here in the community level because I think it's easier to understand the problem. But this qualitatively matches a little bit of the predictions that we have in terms of constraints between different trophic levels. And I guess importantly noting here is that this is the effect of trophic level after correcting for body mass, temperatures, area, and species richness in the community. So those are partial effects. The same thing here in terms of body mass. We had predicted that those effects of body mass would scale to the -1, at least in the most abundant species, and we don't really see that prediction being matched. But at the same time, we see that as you get more and more abundant, then those effects of body mass start to become more and more pronounced. Temperature, interestingly, didn't have any effect whatsoever across the entire quantile range, so it doesn't really matter if you're rare or abundant. Temperature apparently doesn't really have an effect in your population abundance. And maybe the reason for that is related to the fact that we have mostly tropical communities, so it doesn't really fluctuate much. Maybe adding temperature, rocky reef communities would change this. We would have to see in the future. And this is actually the most surprising result to us, and this is community species richness. That was actually the strongest predictor that we got out of this analysis. We did predict it to be -1, and is actually, in fact, is statistically not different from -1 from that equation. But very importantly, it has its effect is way stronger than we would thought compared to body mass, for example. And this is area, and that's actually really nice as well. This is controlling for the effect of sampling. We know that if you're very rare, area is really, really important, right? So if you sample more places, you're finding more and more rare species. But as you get very, very abundant, then area doesn't really matter. It's getting very, very close to 0 because you're everywhere. Unless you're a very clumped, abundant school of fish. But on average, that seems not to be the case. This is a graphical representation of those things. So this is looking at the effect of species richness after controlling from everything else. So going from the species poor community to the species rich community, this is what that -1 slope at the most abundant populations mean. This is statistically showing how would be predicted your maximum abundance to be at very low rich communities and species rich communities, basically. So there is a decrease in your maximum capacity of population abundance. And this is somehow comparable in magnitude the effect of trophic level. After controlling for everything else, this is how you go from, say, herbivores and omnivores to piscivores and carnivores. So the interesting thing here, as I mentioned, is the effect of species richness. We don't really understand what's going on at the moment, but this is suggesting to us that these regional effects in terms of speciation, defining the meta-community and the species pool, were having a tremendous effect in how the species are partitioning their energy at specific communities and populations. We don't really know if this is really dictating competition. It seems like it is, but could be something else that we haven't thought of. We do see these energetic constraints of body size being pronounced towards more abundant species, which is really interesting. But at the same time, they do not quantitatively match our predictions, suggesting that there are many more ecological processes going on that are more important than the energetic processes at this population level. But I guess the take-home message of this section is that we can, using this analysis, we could characterize that you can be rare in many, many ways. If we look back to this plot, so as a small species here can be anything. So it can be very rare in many, many ways. But in order to be very abundant at the very top of this constraint envelope here, you need to be a small species in a low trophic level, and you have to inhabit the species food system. That's what basically the model is telling us. So now scaling up to the community level, the idea of what motivated this section in particular is the idea that we're going to have there are changes in temperature and net primary productivity predicted to occur within the next couple of decades. And they tend to be very dramatic, in particular in the tropics where you're going to have a bigger stratification of nutrient recycling due to net primary productivity. The thing is, how is this going to affect the amount of energy these communities of reef fish can flux? The short answer is we don't know, and the reason why we don't know is because we don't really know what it is currently. So that was the big objective of this chapter, is trying to characterize energetic fluxes of different communities using that same data set I just showed you. So going back to this individual metabolic rate equation, the same idea. You have a bunch of individuals in a community, you're just summing the total, I'm going to call it the total respiration for that particular community. And this is a conceptual figure that is, I think, really important to understand. We have this scenario where, let's say, it's supposed to mimic a pristine system. You have big sharks, you have big groupers, and the different blue things here are just representing their mitochondria, which is a surrogate for the respiratory capacity based on their body mass. So this is the total standing biomass. This is what we call the size-corrected biomass, because this is being scaled to this alpha exponent here. So it's accounting for their energetic capacity. And this is their total respiratory rate. The thing is, we can have a separate community at this exact same temperature with almost half of that body mass, the total standing biomass, but with the exact same capacity of respiration. So this is really important, because it's not to say that if you cut down a lot of big individuals, that doesn't mean that the small individuals are not fluxing that energy. And the same if we increase the temperature. So going from 20 to 28 degrees, using the parameters we estimated from the individual level model, you can have this scenario where you have half of the individuals at this community, but because they're fluxing twice as much energy, they're actually respiring exactly the same thing across all the three different communities. So this is something that people didn't really talk much about in the literature, and we thought was actually really important, because this is the currency that is telling us the effects of environmental changes in respiratory capacity in communities. So going to predictions for this particular section, the first thing is we consider that those energetic constraints going from the base of the food chain to the top of the food chain. So the total production has to be bigger or the same as the total respiratory in a given community, otherwise it doesn't make sense. You cannot eat more than what is being produced. So we recognize this by saying, okay, the total respiratory rate of a community is a portion, is a fraction of that total production. And by modifying that equation, the initial equation of the total respiratory capacity in the community, sorry, another big jump, you get to this explicit expression about how the size-corrected biomass will change if you change, for example, the total production in the system. So if you hold everything else constant in this denominator and you increase the net primary productivity, then you expect the total biomass in the system to go up. And again, the same, if you're increasing temperature, holding production constant, then you expect the size-corrected biomass to go down. So these were the specific predictions we tested for this, using, again, the same database, but now exploring at the community level. And what we did here is we looked at the planktivore section of the database, and the reason for that is we only have a good handle on near-pelagic net primary productivity. We don't really have very good estimates at global levels of reef benthic production. It's much more complicated to get at. So to be fair, we estimated the effects based on near-pelagic net primary productivity obtained from SeaWift's satellite measure. So as we can see, we have, for the productivity expectations, we have a predicted slope of 1, and we got something close to 1.74, but that is not statistically different from 1. So quantitatively saying, this matches the prediction from the theory, but this, really importantly, as we increase the temperature in the system, then holding everything else constant, this is actually holding productivity constant, you see this decrease that is systematically larger than the negative 1 predicted from the theory. So there's something else going on as you're moving from a low productivity system to a to a low temperature system to a high temperature system for these communities, just looking at the planktivores. So we have this bottom-up constraint being manifested by looking at this in this way to the community. We can validate a mechanistic model explaining these broad patterns at broad spatial scales. Maybe the satellite estimate from the sea waves can be a good proxy for the plantivores. We don't really know. It seems like it's okay, but I'm not going to show it here, but at the total community level, if you put everything together, it turns out that a lot of those communities are respiring way more in terms of energy than is being produced by the system, so those numbers don't match. And this is just saying that we're only accounting for reef fish. The system is made up of a bunch of things, you know, a lot of invertebrates and a lot of other organisms that are also respiring and taking energy in that system. So this is to say that probably those estimates are really, there may not be good proxies for total reef productivity. So we really need to get at estimates of reef benthic productivity in independent ways. This takes me to the second prediction. This is again going to the trophic level constraints. We expect the total respiration of herbivores to be 100 times larger than the piscivores, considering a 2 trophic link difference between them. We know in reality this is not a straight linear relationship, but again, it's an initial prediction. And the funny thing, this goes completely against what has been described for pristine system, where you have inverted trophic pyramid. You have a lot of sharks, a lot of groupers at the top of the pyramid, and then you have a much smaller base. The thing is, the authors in these papers, they're trying to explain this by saying, okay, herbivores may have a faster turnover. They die younger and they keep turning over very, very fast, and the long-lived sharks and big groupers, they keep eating them and they accumulate that biomass. But it turns out that these rates in biological times can also be explained by three-quarter power scaling. So if we account for that mass correction that I did for the biomass, we are indirectly accounting for these turnover rates. And then we can see whether these pyramids are going to change or not. Because if this explanation is correct, then that's what we should see. So what we did here, this is just showing the relationship. This is like size, this is standing biomass, I'm sorry, and size corrected biomass for that particular, for different biogeographic regions. And I'm going to take one specific case as an example. So looking at the data we have from the Cocos Island in Costa Rica, you have a 47% of total biomass of piscivores in that particular island. When you correct for body size, you actually get at a 32%. So this is yet very, very high from what would be expected after size correction. So this leads to the conclusion that other authors are also getting at, is that these reef ecosystems are being highly subsidized in terms of energy. There are external sources coming from other places, and in the case of piscivores, they're not limited to one reef, right? So they roam around maybe thousands of kilometers, we don't know, but some estimates actually point to almost 2,000 kilometers one individual shark. So they are going across many or hundreds different reefs, and they're getting energy in each one of those, or maybe even outside the reef ecosystem to the big, deep parts of it. We don't really know, but this is probably accounting for the fact that we're seeing much more energy of going to the piscivores than would be expected if the reef was a very closed system. So, yeah. Size correcting is important, and I'm going to give another example why this is really important. So in that same island, you see this is the total biomass we estimated, standing biomass. So this is 5% of the total standing biomass in the system attributable to the hammerhead shark. And then we have 1.86% of the total biomass attributed to the small dämsel fish. But after you size correct it, taking it to the three-quarter power scaling, it turns out that the size corrected biomass of this guy only represents 2.5%, whereas this guy goes up to 5%. So it's just to say that these small fellows here are respiring way more energy than would be predicted if we were just looking at the total standing biomass in the system. As conclusions for this section, I would say we need to better understand the sources of NPP on reefs. We have other confounding factors that we couldn't unfortunately measure, and those are mostly related to overfishing. So the idea is that many people would think that if you remove those predators, then the mesopredators will grow and become more abundant and so forth. But there are some very recent evidence showing that if you increase human impact in islands in the Pacific, it turns out that the bacteria starts to turn over more and more energy. They're taking that fraction from the fish. It's not actually going to the bottom trophic levels. It's just going to something completely different. Maybe the detritivore cycle, we don't know. So this, I think, in my opinion, this has huge implications for the way we see trophic cascades. And I'm going to close with this section that is going up to the ecosystem level, and this is something that is being in preparation at the moment, and is looking at the effects of growth rates on the trophic, or sorry, the transfer of energy across different trophic levels. So we start from this idea that your total metabolic rate as an individual is being partitioned as the energy that you spend to grow in biomass and the energy that you spend to maintain your own body mass. So it's a dichotomy between maintenance and growth. So we can manipulate that equation in particular here. So you're growing, you're getting bigger, and you get to this expression. This is an ontogenetic model for growth that has been described more than 10 years ago. And what I want you guys to take from this is you consider how you grow in mass across time, and it's this classical sigmoidal curve. You reach a point where you get to your maximum growth rate, and then it starts decelerating until you become an adult. So you basically stop growing, and then all your energy is being spent in maintenance. Sorry. But what is really, really interesting from this thing here we're observing all derives from this fraction here. This is telling us that at a given point in time, your growth rates have a certain amount of energy from your total metabolic rates that derives from this. So if you're really, really small, your total mass relative to your total adult mass is really small. So this is 0, and this whole thing goes to 1, just saying that your total energy expenditure is going to grow. But as you grow, this fraction starts increasing and increasing and increasing, and this is saying that you're spending more and more energy to maintenance, until the point that you reach a big M here as well, and this whole thing just goes to 0. So basically you don't have any growth. So here in this case, dmdt, so your instantaneous rate of change of mass through time is 0. You're not growing anymore. So to analyze this thing for fish, what we did is we collected 2 different data sets. One is we collected pair estimates of growth rates and metabolic rates. So people would put this individual in a poria, would measure their growth rates through time, and at the same time how much oxygen they were respiring. And then we took a different data set, which was using the von Bernoulli type of equations of growth, which are based on length. This is a bit complicated, but what we did with this data set is, based on the parameters we could get from fish base, we estimated their maximum growth rate at their maximum body mass. So this inflection point here, that's when we estimated that growth rate across all these blue points. So that's what I'm saying here. All the blue points are growth rates estimated at their maximum growth rate, what we're calling the optimal growth rate. The thing is, if you look at these 2 data sets, they're not really reconciling, are they? I mean, the intercepts are different. This guy is a bit lower than this guy. So the thing is, as I told you before, early in ontogeny, this whole thing here goes to 1, so saying your energy is going to grow. But these guys here are at their optimal growth rates, or they're already at this stage of the curve. So they have a maintenance cost that we're not accounting for. The thing is, if we account for their maintenance cost at the optimal growth rate, we can actually reconcile both curves and get to this universal curve of growth rate, where you go from a small tiny larvae to a juvenile tiger shark, which is really nice because this was actually just based on the theory. We didn't do anything with the data. This is just correcting based on what the theory is predicting to us, telling that it actually is going to pointing to the right direction at least. So I'm going to point to a different part of the equation now, which is related to this EM. And this is actually a fundamental quantity I'm going to spend most of my time now talking about. So this EM here is just the amount of energy that you spend to create more biomass in your body. So you're assimilating things from what you eat, but then you're breaking them down in small peptides or whatever, and then you need energy to put them together and build tissue. So that's the energy that is being accounted for. And that EM, in particular, is basically just the fraction of your total metabolic rate that is being allocated to growth when you're young, because your total energy is going to growth anyway. So that particular fraction that we can measure, because we have paired estimates of metabolic rates and growth rates, you can measure that and you can quantify how much energy the different species and different individuals need to build new biomass. So this is the thing here. This is with that compiled data set, and this is with the fish-based data set. They yield relatively close amounts of energy, but this is early on in ontogeny, and this is at their maximum growth rate. The thing is, so this is about 4,000 joules per gram. So that's the amount of energy you need to build new biomass. For each gram, you need 4,000 joules. That's what this is saying. But at the same time, this is on the log scale. You can see there's a huge spread in the data, saying some species need way less, some species need way more energy to build biomass. This has really important implications, and the reason for that is related to this, to the assimilation. So when we eat something, we're trying to build new biomass. All our energy is basically partitioning two things. Is our total metabolic rate, so all we're respiring to produce energy, plus the combustion energy of the biomass you're eating. So if you're eating something that it has calories, right? So there's energy associated with that mass that you also need to take into account. That's your total assimilation rate. Whatever you're respiring plus the energy containing that mass. So the thing is, at a given point in time, you can be predated. And that the energy that is going to the next individual is your body mass plus the combustion energy of your body mass. That's the total energy that is going to the next trophic level, for example. So if you want to calculate the efficiency of a particular individual, basically it's the total combustion energy of its mass at a given point in time, divided by the total amount of assimilation energy the individual has spent across its entire life up to that point. And we do it based on this equation here. We show that there are important consequences of changing this EM as you go. For example, say if you're an individual that needs 10 kilojoules per gram to build new biomass, your efficiency is going to be way lower than if you need only 1 kilojoule. And the reason why this is not linear is because this is in the denominator, so it's a nonlinear function. So it's not really straightforward. And also very importantly, and this is actually really relevant to reef systems in my opinion, is because as you grow older, you're reaching your big M here, your asymptotic mass, and then that means you have been spending a lot of your energy to maintenance. But if you're early on in ontogeny here, very small larvae, then most of your energy is going to growth. So if somebody eats me at that point in time, they're getting a lot. I haven't really spent much energy to the system by respiring that energy out, right? And I believe that may be one of the factors explaining why reef systems may be so economic in terms of energy, because most of them are thought to be oligotrophic systems. And maybe those events of predation, when you have massive spawning events for all the organisms in the reef, are actually telling this. The organisms are taking the best of energy transfer in that system by doing those predation events. So this is, in my opinion, this is really cool, and I don't really see many people talking about it. And also this, we really need to characterize and try to understand how much this energy changes across the different species if we are to understand these ecosystem dynamics of trophic efficiency across the different trophic levels. And probably it's not only to say there's a 10% thing going from the bottom to the top. It can be way more complicated than that. And that's what this figure here is showing. So I'm going to wrap up here, moving forward. So in conclusion, all these different topics here, I think they tell us that general theory can account for many patterns. We saw the effects of size-corrected biomass being constrained by temperature and productivity. We saw that accounting for the maintenance cost of growth, we can actually line up different growth curves of different data sets. We saw that the most abundant species in the systems are getting more constrained by energetic patterns and so forth. But in a technical level, I think that the hierarchical random effects model are also very useful because they allow us to partition that variance in a statistical model that we would say, okay, this is residual variance. But not really. You can actually attribute that variance to the different levels of your hierarchy. So this is really interesting because by doing that, we can actually try and understand, for example, why the different species and why the different families differ in terms of their metabolic rates or growth rates between one another. I haven't really talked about this, but I think this is actually important. The potential for adaptation to climate change. One of the things we did cover in that paper is we provided a model that can estimate how much you adapt to different temperature regimes. The data didn't really provide any evidence for that, but there are independent evidence showing that there may be adaptation. It's again, it's a topic open to debate. We don't really know, but in my opinion, just going again to what I just said, is predicting these trophic web dynamics, ecosystem level dynamics, all go through these energetic processes, and if we don't really understand metabolic rates, growth rates, assimilation, constraints in population, ecosystem, communities, and so forth, we're not really going to understand what's going on with climate change scenarios and things like that. So to wrap up, these are my overseas collaborators. Without them, I wouldn't be able to do any of this. So they have been really helpful, giving many insights in terms of reef-fish community perspective and also the data sets. My supervisor at Macquarie, Drew Allen, who has been the theory guru for me. It wouldn't be possible without him. And finally here at JCU, thanking Tom Bridge and Rebecca, who facilitated this talk. So thank you very much. That's it.